There's that yellow box in the corner that says live. Do I believe it? It worked last week.
I
Anyway.
believe it. I see it.
Anyway, I have two cats with me this week. That's all I could round up. The other cats are missing in action. Hogtie Champ, Jade West, Dragonaut, get your thumbs in here. Anyway, tonight is the follow-up to last week's hangout. Last week's was an overview of what cancer is and how it basically kills you. This week, it's the genetics behind cancer, i.e., what makes a cell suddenly become a cancerous cell. And it's all down to genetics and various switches. Now, one of my poor unfortunate cats, RSC, he's actually pulled together a presentation with slides. And I'm going to turn the floor over to him. And I'm going to let him tell you all about this. We've also got Sam. And Sam will be basically jumping in to say anything that he's to do. I'm basically giving the floor to the guys, and I'm basically letting them get on with it because I'm lazy. So, it seems that Ruhif is saying, turn yourself up, Fiona. Is that any better, sweetie? I have no idea. My microphone is not doing what it
You're
should.
coming in a little soft.
Yeah, I was going to say a little bit soft, but I didn't know how it sounded to the outside, and also I'm on my phone, so who the hell knows what's going on with the phone app.
Let me just check. It used to do this, then it stopped doing it, and it started doing it again.
All right, give me just a little second. Give me a couple minutes to download the presentation because it's unfortunately.
Yeah, and hopefully, RSC, if you can peep at the external chat, I linked that very useful diagram that we were talking about, where they, it's basically it's called the subway map of cancer.
Is that any better?
Much better, at least to me.
Right, well, all I did was roll the little roller backwards and forwards and put it back to where it was. I must have, what is it? Somebody told me dirty pots. I've got dirty pots. Right, anyway, so people, I am going to hand this over to RSC, and he is going to go through this, and Sam can leap in whenever he has to. So I'm going to present RSC to everybody, and I'm going to mute my microphone. Right, off you go.
Let's see how well this goes with the presentation. Sharing. Fiona, can you see my presentation?
Right, I can see that. Can everybody in the outside chat see the slide on the screen? And does it stay there when I'm talking?
It is for me, but I know that I'm not on the outside.
I'm trying to look on a secondary device, but it is not working well
everybody
either.
says it is. Everybody says it's staying on. So I'm now going to mute RSC. Break a leg.
Well, thank you, Fiona. And thank you, Sam, for joining us. Welcome, everybody. Everybody can call me RSC. You see my name up there on the screen. Just a disclosure, just to let you know a little bit about myself. I'm an MD. I currently am in the Cleveland area. My primary practice right now is medical research. And again, this presentation is solely for educational content and information. I don't want anyone to take the words that I say today and use them as the sole opinion on making a medical advice for themselves or people that they know or care about. So if you do have anything that's related to our discussion today, please make sure that you are talking to the right people. That would be your physician, your primary caregiver, and the people they put in the team to take care of you and your problems. I don't have any financials. I don't own any stocks in medicine or anything like that. There's really nothing for me to gain from giving this topic as a conversation or a presentation, other than just increasing knowledge and education for everybody. So the topic today is genetics and cancer, and here's a little bit of an outline of the things I wanted to cover today. I understand that there's a lot of confusion with a lot of the terms that are used, cancer, genetic terms, so I want that to be the first section we go over. And Sam, if you want to jump in, help me describe these terms so we can have everyone in the audience understand them fully. I'm going to make it my goal today to use plain language, communicate in a way that makes sense for everybody, regardless of your background, whether you have a PhD like Fiona, or whether you're a high school graduate, or whether you're still in high school now. My goal would be to make this education and this material as easy to digest for everyone as possible. After we introduce some terms, I'd like to go into how cancer develops. And some of the main topics in this section would be damage to DNA, discussing what are proto-oncogenes and oncogenes, and then a brief section about epigenetics, because that is kind of an indeterminate player in all of this. It's not certain at what extent that has, but I think it's worth discussing. From there, we should talk about the specific genes and how they contribute to cancer. There are a lot of different hypotheses out there, and they are, at this time, not proven. They're not theories like you would call germ theory of disease, or evolution, or the theory of gravity. But they're ideas of how cancer develops and why cancer develops. And I think it'll be nice to go through a couple of the current models that science is investigating, that medicine is investigating. And from there, we're going to go and discuss some specific examples of genetics. RB, the retinoblastoma gene, P53, and the BRCA mutations that people are aware of that cause breast and ovarian cancer. So if there aren't any questions, I'm going to continue. So I want to make sure I'm on my first slide. So let's talk about some cancer-related terms. Chemotherapy is the first term I can think of. Chemotherapy is basically a word that is used to describe any kind of drug, any kind of medicine, any kind of therapeutic that's given with the purpose to kill cancer cells. Hopefully it's a very understood definition at this point. Let's talk about malignancy and malignant. That is a term that's used to describe tumors that are considered cancerous. We'll go through the difference between tumor and cancer in a little bit, but I think explaining these terms first will make it easier for everyone to understand and keep up with. A malignant tumor is a tumor, or refers to a tumor or a cancer that is able to spread to other tissues, to invade, to go and affect other parts of the body, not just the local area where it's forming. Metastasis. This is the spread of the cancer, when a cancer has spread from its site of origin to another area in the body. The second portion of the cancer that has spread is called the metastasis. And you can read the definitions here, but a majority of metastases occur in brain, bone, liver, lungs, places where blood and lymph system tend to connect and gather fluid together. That's just where they're going to travel. So let's talk about tumor. I've actually described tumor twice to emphasize the point. It'll be on the next slide as well. A tumor is any mass that forms when you have abnormal growth. So tumor just refers to any mass. Some of you may have had benign tumors in the past. A lot of children are born with little red bumps on their skin. Those are tumors, technically. They're benign tumors. They don't spread, they don't invade, they don't attack other areas, but any area of growth that is not normal is called a tumor. It could also be called a mass or a nodule or a neoplasm as well. From there, we classify tumors as either benign or the malignant. Benign meaning not cancerous, not spreading, not damaging, basically not cancer. So some other terms that will be helpful as well. Adjuvant therapy. This is treatment that is added in addition to the main cancer treatment. Its main goal isn't to kill cancer cells. Its main goal is to prevent cancer from coming back. So you may hear about this. This is one of the new ideas in medicine, is that not only do we need to give something to treat the cancer, but we want to keep the cancer from coming back. So adjuvant therapy is a very new thing. Lots of research, lots of new medicines are coming out as adjuvant therapies. And that's what they're talking about. They're not talking about the medicine that's going to make the cancer shrink, going to make the tumor shrink, that's going to wipe out the cancer. They're talking about therapies that keep the cancer from coming back. A biopsy. A biopsy is a small sample of tissue taken from the area of a tumor or cancer. Biopsies are studied by pathologists in labs where they will take samples and cut them up into hundreds, if not thousands, of slides. And they will go through slides for hours and hours to examine for differences in cell types. This is actually very difficult to see, and I think you'll come to appreciate that a little later with some of the pictures we have to show. Carcinogens. Carcinogens are any substance that can cause increased risk of developing cancer. These can be things like tobacco smoke, formaldehyde, even some medications can cause you to have a higher risk of developing cancer. Carcinogens don't mean you're going to get cancer. It means exposure to them will increase your risk for cancer. That's as far as anyone can say about something like that, because we all know lots of people who smoke well into their 80s and 90s who never get lung cancer. And we know some people who never smoke who get lung cancer. Carcinogens can only increase your risk. It can never be determined that you're going to get cancer from smoking. Remission. Remission is the goal for cancer therapy. Disappearing of cancer symptoms and disappearing of the mass, disappearing of the positive test results, that is the therapeutic goal that oncologists seek. They realize that it's impossible to say cure, especially in this day and age where people live so long that cancers tend to come back if you live long enough. For women, breast and ovarian cancer seem to be part of the aging process almost. If every woman was to live till 150, I would expect to see 99% of them getting breast or ovarian cancer. For men, it's prostate cancer. As men age, your risk of developing prostate cancer goes up, almost to the point of being 100% at the age of 100. It seems to be just part of the normal aging cycle for humans. So again, let's go through tumor one more time. A tumor is a commonly used non-specific term for a new growth. It just refers to a mass that's abnormal. It can be in any part of the body. Now, most of these are classified as either benign or malignant growths. Sometimes you don't know whether it's benign or malignant, but we're going to keep it simple. Benign means harmless. Malignant means cancerous, dangerous. All right, so now let's talk about some genetic terms. Sam, if you want to jump in here at any point, feel free. Allele. These are just alternate forms of one gene. So when we talk about alleles, we're talking about a gene and the different types of genes that are available at that location for the DNA. An individual who's labeled as a carrier has two different alleles for a single gene. Usually in a carrier, we're referring to one allele as a diseased allele and one allele as a normal allele. Chromosomes. These are the gene-carrying bodies for the cell. They carry long strands of DNA that compose millions of base pairs. They're made up of the nucleic acids incorporated with protein, wound around in double helix, and humans have 46. Evolution, a term hopefully we've all come to know very well. Genetic change in a population of organisms that occurs over time. Genes, these are the individual units that are inherited, and they occupy specific locations on chromosomes. Each gene is a sequence of DNA, and that is the key. It's a sequence, a specific sequence of DNA. And that sequence will be encoded into an amino acid through the process of translation, which is the making of amino acids from DNA. Some more genetic terms. I know it's getting kind of boring. We haven't explored a lot yet, but genetics is the study of genes. Genotype is the genetic characteristics, the genetic sequence of an individual. It's all their genes added up into one sum. Phenotype is the observable, is the outward appearance of that organism based on its genotype. So I don't know if we have any questions at this point, but any questions in the live chat now before proceeding?
None so far.
Okay.
There's been a couple of questions, slightly not about what you're talking about. I'm dealing with them as we go along.
All right. Well, I'll keep going.
Yep.
Please button if there's an on-topic question. Let's get those answered, and while we're in this section. So let's talk about what cancer is now. Cancer is actually a broad term. It's a collection of diseases, and in all types, it begins with the cells of the body dividing in an uncontrolled manner. These cells go on to divide and spread and eventually cause defects in the body, in the organ, in the tissue that they occupy. So cancers can start up anywhere because your body is made up of trillions and trillions of cells. They'll grow and divide just like any cell, but at some point they either acquire some kind of damage, some mutation, and instead of dying like a normal cell would, they continue to replicate and make new cells and copy themselves. And so if you want to see some of this information, you can see the government website I've put down there. A lot of the information and pictures I've gotten are from that website because I felt like they did a very nice job at explaining cancer. The tumor versus cancer, what is the difference? So I said cancer was a very broad definition or a very broad term. Think of tumor as an even broader term. And cancer is a subset of tumor, that in which case not all cancers are tumors. So like I said before, tumors include any mass, lump, lesion, neoplasm, any abnormal cell growth. The benign ones don't invade, they don't cause problems, they stay where they are, they'll grow to a certain size and they'll basically stop. They won't cause problems. Malignant tumors are the ones that can spread, that can damage tissue, that can travel to other parts of the body, and these are cancer. But not all cancers are tumors, because like I said, tumors are solid masses. So if you think of a cancer like leukemia, leukemia is in the bloodstream. It's not a solid mass. It's not a solid tumor, but it is a form of cancer. So this is where the distinction comes with not all cancers are tumors. In some instances, there are benign tumors that can transform into malignant tumors. However, we're very lucky. This tends to happen over long stretches of time. So if you can think of one example, maybe you've heard of someone in your family who's had a colonoscopy and they find out that they have a polyp. Well, a polyp is just a tumor of the colon. It's a benign tumor for the most part, unless you have some sort of colon cancer, but they will tend to transform into cancer over decades, over years. Not anything that would happen quickly. So since we know that there's a chance that these kind of benign tumors can transform, the doctor normally just cuts those out immediately. Why risk it? If the risk is more than 0%, let's just cut them out because it's a very simple thing to do. And that's what they do. And then they'll go back and you'll come back and be seen for colonoscopies again in the future to make sure they've caught everything. So again, I want to emphasize one last time that tumors are a big category. It's a catch-all term for any kind of mass, lump, lesion, neoplasm, abnormal growth in the body. It has to be a solid, a contiguous abnormal growth. Cancers are a subset of tumors that do include things like leukemia, but the things that bind all cancers in terms of definitions are the ability to invade, the ability to attack other tissues, the ability to travel to other sites in the body. So I think this will be the last slide where we're talking about definitions. We'll get to see some pictures and hopefully have a conversation with Sam and Fiona. But let's talk about cancer cells versus normal cells. Why is there this big difference? Now, cancer cells are different from normal cells in many ways. And the most noticeable one is how they grow, how they invade, how they behave. Cancer cells do not function in the same way that normal cells do. Normal cells will go through an aging process, they'll mature, they'll eventually die at some point. They're programmed to go through this life and death cycle. Cancer cells do not follow that cycle. They ignore the signals that normal cells hear and respond to that tell them, okay, now is your time to stop working. Now is your time to retire and eventually they go through a process called apoptosis, or programmed death. Cancer cells completely ignore all of that. Cancer cells also influence normal cells and normal molecules and blood vessels. They tend to pump out hormones and give out signals to the body indicating the need for blood, for different nutrients, for oxygen, because they need all those things to survive also, and they are very greedy if that can be used applicably. And the final thing that differs is that cancer cells normally evade the immune system, which means there's a system in your body that is designed to detect cancer and erase it from your body. But for some reason, they've come equipped with a way that prevents that. And that's why it's so difficult to tell them apart from regular cells. They are basically sheep's and wolves, or they're wolves in sheep's clothing.
I see. I can elaborate on that a little bit if you want me to.
I would love for you to.
So what you can imagine is that your cells are constantly presenting pieces of proteins on their cell surface so that you know that it's a normal cell. And if there's abnormal proteins, the cells will put that on the surface, almost like holding up a sign like, hey, it's abnormal, and an immune cell will go by and kill the cell, because obviously you don't want, just abnormal cells floating around in your tissues. They could be potentially dangerous. One of the big things for cancers is that they will find ways such that they evade this type of detection. And because they evade that type of detection, they're able to keep going. And so you don't have the action of the immune system to kill these cells. I don't know if that's a good analogy for how that works, but that's my understanding of it.
I like the analogy, Sam. Do we have any questions in the live chat yet?
I'm looking now.
Yeah, I haven't seen any coming up. I think so far everybody is following along fine.
Okay,
remember,
good.
if anybody doesn't understand something that's said, put it into the live chat, and then we'll stop and we'll go back to that and clear it up for you, because we don't want to keep all of this to the end. We want you to be able to follow along as we go. Okay?
Definitely. Thanks, Drag, for participating. We appreciate it.
Yes.
Yeah, my throat's kind of shot, so I'll participate where I can.
All right.
Cool.
Okay, so now we get to actually go into some visual things. So take a look at this slide. I did not put this slide together, so I'm not taking credit for it, but this was a very nice slide that I found online. And you can visibly see the difference between the cells that are normal on the left versus the cells on the right that are cancerous. With the right kind of staining, with the right kind of reagents, we can entice this kind of picture when we look at cells. So I talked about biopsies earlier. This is the type of thing that a pathologist will look for. They'll look through millions and millions of cells to find just a couple of cells that look like the cancer you see in that right column. The body doesn't have eyes. The body isn't visually looking at the cancer cells, the normal cells, like the way we do, or the way that the pathologist does when they're examining a biopsy. Like Sam said, the cells basically smell or feel each other out based on the proteins they have on the surface of the cell. And cancer cells do a very despicable thing where they have problems that are inside the cell, but they don't express them on the outside of the cell. So the body isn't able to detect that those problems exist inside those cells, and they go unchecked, basically. So you'll see a lot of characteristic differences is the cancer cells will tend to have very, what are called dense and dark nuclei. This is the darker spot in the middle of the cell where all the chromosomes, where all the DNA is housed. Also, you'll tend to see cells that are rapidly dividing. In the second row, the cancer cells, you'll see their nuclei, their chromosomes are the dark little blobs in the middle. The reason they're so dark and condensed is because those cells are dividing. They're cutting them, dividing into two, segregating their chromosomes into making two daughter cells. And they're doing that so regularly that the chromosomes don't have a chance to unwind and fill up the entire nucleus and give it that normal look that you would see on the left. Also, something you'll see in cancers, even though their proteins on their surface will be expressing things that are the same as the normal cell expresses, their shape will be different. But the problem is the immune system is unable to recognize shape. Cell shape is not something that it uses as a criteria to identify cancer cells. But luckily, the doctors can use that as a characteristic to identify them. And also, you'll just see very weird features sometimes in cancer cells, like on the bottom row. They will look extremely abnormal, and sometimes it's very easy to see on a slide. But sometimes it's so difficult to see that it takes decades to train your eyes to find this kind of stuff. And it takes a very nuanced education to appreciate the skills that we have now, the materials, the techniques we have to identify these cancers, because these cancers have been around for as long as humans have been around, most likely. We just have been unable to see them and appreciate them until now. So this is a nice diagram I also pulled up that kind of shows the difference between normal cell division on the top and cancerous cell division. On the top, you can see that you have one cell that divides into two, and then the cell that has already divided once divides again, but one of its daughter cells gets some damage that it can't repair, some kind of insult it can't overcome. And it goes through the normal process. If cells can't repair their damage, they're supposed to go through this apoptosis, this cell suicide. This prevents cancer. This is a way for the body to control and make sure that only normal, healthy cells continue to grow. But in the bottom, you'll see that mutations can accumulate that aren't repaired in cells. And these mutations can build 1, 2, 3, 4 on top of each other. Not all at once. This likely takes a long period of time. Don't be fooled by the fact that it says first mutation, second mutation, thinking that this happens chronologically one after another. There are likely long spans of time in between each of these incidences. This is just for visual sake. But eventually you get to that one critical mutation that puts you over the edge, where you've gone from a normal cell that functions normally to a tumor, to an uncontrolled growth that does not function normally anymore. When will that become cancer? That depends on the type of tissue, the type of tumor, and all the types of mutations that have occurred previously. So here is a nice breakdown of how cells proliferate, how cells grow. We can see that if you start in the upper right-hand corner, you have a mutation that inactivates a tumor suppressor gene. So mutations are detected by the cell. There are several mechanisms that we'll talk about later that tell a cell, hey, you have an error somewhere in your genotype or in your genome. You need to stop what you're doing and fix it. And it's literally a signal that's that abrupt. Signals come through that inactivate, mutations can cause DNA repair to occur, and things like oncogenes can be activated if the mutation is in the right spot. If these kind of things aren't corrected, you go and develop cancer, tumors as well.
Can I cut in for a second?
Yeah.
People have heard Tommy mention oncogenes, and I think, and please correct me if I'm wrong, I think that he was under the impression that they're there to cause cancer, like that is their purpose. And the issue is that that's the furthest thing from the truth. How I was taught was that there's tumor suppressor genes, and then there are oncogenes. The way you can kind of distinguish them is that one's a gas pedal and the other's a brake. So if either one of those is broken in your car, guess what's going to happen? You're going to have a wreck. So that's the way that you can think about it, as a tumor suppressor genes as being the brake, and oncogenes as being the gas pedal. So that's something that I use to help remember both of them.
Yeah, thank you for the great analogy, Drag. I actually have another slide. I think I'll go through that a little bit more here in a second. But on the left, you can see the basics of the cycle that a cell goes through. It goes through phases called growth phases, which are represented by the letter G, followed by a DNA synthesis phase, followed by a second growth phase. And you can see there are several purple blocks in that cycle. These are checkpoints. These are areas where if DNA damage is detected in a cell, there are mechanisms for the cell to halt everything until that damage is corrected. So these are the brakes that stop the cell from going into that proliferative, cancerous, tumorous growth. These phases we'll go into a little bit more in depth later, but this is just a general kind of breakdown slide. It has a little bit more information than I want, but not so great with making images on my own. So there are 5 main types of cancer we can go through real quick. Carcinomas, these are tissue, skin, internal organ cancers. Sarcomas are cancers of the bone, cartilage, fat, muscle. Leukemias are blood and bone marrow. Lymphomas start in the immune system, like your lymph nodes. And there's also central nervous cancers that can develop in the brain, the spinal cord. So here's a nice diagram of where you might find some of these cancers. Bloodstream lymph nodes are everywhere. Fat, bone, and muscle, we're all familiar with where they are. Obviously, colon cancer can only occur in the colon, but if you see colon cancer somewhere else, you know that it's a spread cancer, that it's a metastasis. The prostate is only in one place in men. The breasts are only in 2 places in women. So if you see breast cancer somewhere else, you know where it's come from.
Can I just point one thing out here? You say it says in your slide, breast women. Men can get breast cancer as well.
True.
Yeah. And so all you guys out there in the live chat that have never done this before, get checking yourself, not your other half. Sorry, on you go.
Good point, Fiona. But I actually think that they have stopped recommending self-exams. I don't know. Talk to your own doctor about that, what they want you to do. So here's just kind of a case-by-case list of different known carcinogens. So things like excessive weight and obesity, that has been shown to cause an increased risk for many types of cancers. Certain things in your diet can cause cancer. Tobacco, obviously, is a huge precipitator of cancer. Viruses, alcohol, UV lights. I mean, the list goes on. This is not an all-inclusive list, but this gives you a general idea of the breakdown of the things that can contribute to increasing your risk for cancer. So what are we going to do here? This is a little slide I put up that we're going to take a break. We're going to say, okay, that's nice. We've kind of talked about cancer, what cancer is. Hopefully everyone has a good idea of the difference between a tumor and cancer. We understand where cancers arise, what it means to metastasize. Are there any questions about cancer and how cancer develops at this point before we move on?
Did Trevor have a question in the live chat?
Yes, he does. Trevor's asked, in a benign tumour which appears to not change in size, are the cancer cells changing / multiplying too slow to be obvious, or not changing, multiplying at all? I would say if it's a benign tumour, then by definition it doesn't have cancer cells in it. But we've already mentioned that some benign tumours can become cancerous. The problem with that is that you don't know until you start to see changes in the benign tumour and go along and have it biopsied. But if it's not doing anything, if it's been the same for years and years and years, then I wouldn't be too concerned about cancer cells in the middle of it. Does that make sense?
That was an excellent explanation. That's exactly what I would tell a patient. If you have a tumor, and again, we're talking about a lump, a mass somewhere that has been there since you were born, it may be a tumor that has a chance to become malignant, but the fact that it's been there for so long indicates that it's not as likely. So those are the kind of conversations where you need to talk with the person who's managing your care and say, what is my risk for this becoming malignant? Is it worth it for me to have it removed, or is there no benefit? Because there are some cases where you remove a benign tumor and it doesn't offer you any enhancement in reducing your risk for developing. So,
Yeah,
if I could just, what's the point?
Yeah, if I could just say at this point, my son has a mole in his neck. And it started off when he was a little baby, just a red mark, but it is now a proper mole, a raised mole. And I just so happened to look at his neck one day, and I thought the mole had changed. It looked like it had a darker ring of flat skin around the mole. And I had him straight to the doctor. And he had to go to the hospital, and they photographed it. And then he was back again in 3 months, more photographs taken, then back in 6 months, more photographs taken. So they could see if it was changing. Do you know what that dark ring around the mole turned out to be? Dirt. He wasn't washing his neck properly. And it was because the mole was sensitive. So he didn't rub there. But as soon as I saw what I thought was a change, that was it. I went, but there you go, dirt. On you go.
Well, that's probably the best news to get. That it wasn't anything serious. But Trevor, back to your question. Whether tumors are necessarily growing or not, a lot of the times tumors reach a stage where the mutations that have built up have gone through a course, where the ones that were so unfavorable have caused those cells to actually go through apoptosis or be identified by the body to be killed, and you just have a mass of tissue that isn't necessarily in the growing phase and isn't necessarily in the phase where it's going to die. And those are a lot of the times where tumors come to like a resting phase, where they don't grow, they don't change. There's not going to be necessarily more growth or more cell division in that kind of a tumor than you would find in an actively growing tumor. But it's not the same to say that there's no cell division, there's no growth at all. There's probably just enough growth to maintain the size, the shape, the structure. And a lot of that's going to be based on the cellular environment. Basically, the point when a tumor stops growing, it's dug its own niche, and it doesn't go beyond that niche, doesn't stretch beyond that at that point. Because again, it's not a cancer, doesn't have the ability to invade and go other places. And it's, for some reason, that growth factor, whatever mechanism it was in the past, is likely no longer playing an active role. What it is for each case, each tumor, I don't think we have a good answer for you at this point. But that would be the general thought, is that for whatever reason, we probably aren't going to be able to identify or tell you it's slowed down now. And that's generally a good indication. You don't want growth to keep getting bigger and bigger. That's generally bad. Was there anything else? Any other questions? I thought that was a great question from Trevor.
I think it was Exile was asking if, what did she say? If hemorrhoids were cancerous, then I think it's, I just lost it.
Yeah, she was asking if they could become cancerous. I would say highly unlikely. They're varicose veins.
Yes. So hemorrhoids aren't actually tumors. They are just outpouchings of the veins that run inside the anal canal. Basically, that is an area where muscles cross over skin, and there's not a lot of protection. There's not a lot of space between the end of those veins, the walls of those veins, and the skin, the anal canal. And so what happens is, if you get a lot of back pressure in your veins, increased pressure of the blood system there, vein walls are very weak. They don't respond well to higher than normal pressures.
Yeah.
They will balloon out. And that's what happens with hemorrhoids. They're just balloonings of those veins, which is why they bleed so.
Sorry, pressures like pregnancy And a
Vice
56-hour induced labour. That will do wonderful things to your hemorrhoids.
Yeah.
Yeah, I think you're
Friction is-
talking from experience. Are you, Fiona?
They'll be hanging down to your knees before you know it.
And so
one
that's
of
also
the
another
other
reason.
things is
Go ahead,
go
Drag.
ahead. I was gonna say one of the other things is friction too. Friction in that area definitely cause some hemorrhoids. I know from personal experience. Driving for 18 hours was a very
Yeah.
uncomfortable seat.
Sitting for long periods of times has been shown to increase your chances of developing hemorrhoids. Also, bodybuilders who do a lot of squats, who do a lot of deadlifts, that puts a lot of strain on your abdomen. When you strain the abdomen, you're increasing the pressure in those veins. The place where they're going to pop first is the hemorrhoids. And so I've seen a lot of very fit individuals on their knees crying because the hemorrhoids were that bad, and they can be that bad. But they're not cancerous growths. This is not abnormal cell growth. This is a defect basically in your vasculature. Your veins are ballooning out, unfortunately. But a very good question, because not everyone can appreciate that. Anything else we can address?
We've got somebody who's got a bump in their finger saying it's been there for a long time. Should they have it looked at? If they're worried, yes. If you're worried about it, then...
Well, yeah, the first question I would ask is, what happened? How did you get the bump?
Yeah.
Did you cut the finger and then you got a bump? Or did the bump come out of nowhere? The more worrisome type is the bump that comes out of nowhere.
Mm-hmm.
People frequently get these tumors called keloids whenever they have an injury or a cut in the skin. Those are tumors, but they're benign tumors. They confine the area of where the wound was. They never spread. They never become malignant. Those aren't the kind of bumps to worry about. The kind of bumps to worry about are the bumps that come suddenly, that grow large quickly over like the course of months. And they come in areas where, hey, I get a lot of sun exposure. That's always a risk factor for cancer. And I don't know if there's any other things that you might know of that could lean towards cancer coming just in the skin, but UV light is the number one. Okay, so if there are no other questions, I'm gonna go on and we'll move on to the next subjects. All right, so how cancer develops. So we've talked about what cancer is and how it develops. Let's talk about how genes and genetics plays a role in that. So the basis of cancer is it's a genetic disease. Occur in genes that cause cells to grow and divide out of control, because the controls for growth and division are in the genes. Genetic changes can be inherited from your parents, or they can arise during the patient or the person's lifetime as a result of mutation, errors, damage to the DNA. Every person's cancer is unique. It's a unique combination of genetic changes. The changes in one type of cancer may be the same genes being changed, but likely the sequence is different for each individual. So in general, cancer cells have many genetic changes, mutations in the DNA that go unrepaired, that go unrecognized. And these changes may or may not have anything to do with cancer. But cancer may be a result if these accumulations occur and they're not checked by the immune system, not checked by the DNA repair mechanisms, not checked by the normal functions of the cell. So here's a nice little diagram of how this kind of happens. And it's not technically like it's drawn. The lightning bolt is just an indicator, an animation for DNA damage. DNA damage can happen through any of those carcinogens we listed previously. DNA damage can also happen spontaneously. The molecular mechanisms in the body that replicate DNA, they have an inborn error rate. They incorrectly copy the DNA from time to time. And I mean, it's, I think, 1 in 1 billion, but you think about your DNA in each cell contains over 3 billion base pairs. There are going to be 3 errors every time you replicate a cell, you make a new cell. So that will add up over a lifespan. Think of the number of divisions, the number of years, the number of decades. And you can see how through great lengths of time, one little damage, one little error in one cell can cause many cells to become mutated. And those mutated cells will have a higher risk for becoming tumors in the future. And once they become tumors, they could progress to cancer in some instances.
If could I just pop in here just to say, see that picture you've got there? Right. So we've got the middle one where we have one little cancer cell there. And then we have the one on the right where there's a number of cancer cells. Now, that's come from the multiplication of the original cancer cell. It's not that the cancer cell infects surrounding cells, so to speak. Fair point?
Yes, thank you for clarifying.
It's just because some people, just because of the way the diagram looks, it looks as if, you know, one cell's been called cancerous and it causes its neighbours to do it. It doesn't. That's the cancer cell is multiplying.
Yeah,
Okay,
and I didn't mean for it to come across that way, but unfortunately, that's one of the limitations. Any good drawing you use, different people are going to interpret that differently. So I want to thank you for clarifying that, because that was not something that I was thinking about when putting up that slide. So I think this slide here kind of elicits out your point that you were making, Fiona, that it's not the fact that the cells are spreading their mutations to neighbors. It's the fact that over time, one cell is going to go on to give rise to future cells, and those future cells will carry on any mutation that's previously occurred. I think this diagram does a little bit better job of demonstrating that. You can see as you go down from row to row that one cell gets the first mutation, and that first mutation is then passed down to daughter cells below. And those daughter cells can individually acquire their own unique mutations and pass them on to their progeny, basically, until the point where so many mutations have collected that malignancy, that cancer, that a tumor develops, that cell growth becomes uncontrolled, that all the mechanisms we talked about previously have become enhanced in one cell line. And that line of cells grows, multiplies, and begins to function as a cancer at some point. So the drivers of cancer, and here's where I think we'll get into more of the definition of proto-oncogenes and tumor suppressors. Genetic changes that contribute to cancer come from, I wanted to break it down into the 3 lineages that we talked about earlier: proto-oncogenes, tumor suppressors, and DNA repair mechanisms. People have called these the drivers of cancer. And Sam, I don't know, this was a point where you made a note. You want to jump in here? Okay, I'll take that as a no. So let's talk about proto-oncogenes. Proto-oncogenes are normal genes in cells. They allow for normal cell growth, normal cell division. These genes can become altered, and altering these genes will likely lead to higher rates of cell growth and cell division, and hence they could become cancer-causing genes. When proto-oncogenes are mutated in a fashion that tends for them to be cancer-causing genes, we change their name. We call them oncogenes now. Oncogenes allow cells, cancer-causing mutations, to grow and survive when they should not be growing, when they should not be surviving. Tumor suppressor genes are a different mechanism for the cell, completely independent of proto-oncogenes. They also control cell growth, cell division. These, in cells with certain changes to tumor suppressor genes, division goes uncontrolled. If you remember the cell cycle I showed you, tumor suppressors are responsible for one of those breakpoints where the cell cycle stops, where the cell stops the synthesis of DNA or the growing phase and goes into a mode of repairing DNA. And if the DNA doesn't get repaired in the appropriate time, the cell goes through apoptosis, kills itself to remove itself from the gene pool, basically. The next driver, DNA repair genes. These are genes, proteins function to fix damaged DNA. Many of these genes exist. I don't think we've actually discovered all the genes that are involved with cancer repair. But once you damage these repair genes, it's very easy to see how mutations are more easily acquired because you become deficient in the ability to repair DNA in those cells.
Hey, RC, Sam can't speak right now, so he's sending me messages. Basically, he wants to say that oncogenes tend to be growth factor receptors, and things that cause cell division. An example of this is EGFR.
Yes, Sam's right. Many proto-oncogenes code for things that cells require, that are signals for the cell to grow and divide normally. When they become overexpressed because of mutation or changes in the genes, they are functioning in a way that tends towards developing cancer. And then we start to call them oncogenes because they're no longer functioning in the way the proto-oncogenes generally function. And EFGR is a very popular one. It's been well studied. All cells have receptors for this specific factor. And when it is the appropriate time in that cell cycle diagram, the factors are made from the DNA to tell the cell it's time to go on to the next phase to start growth and replication. And the cell requires these factors to be to go to the receptor in order to initiate and continue the growth cycle, the synthesis cycle, and so on.
So he's saying that epidermal growth factor receptor, that's the EGFR, tumor suppressor genes tend to be DNA repair genes as well as cell cycle genes. An example is caspase 8. It usually triggers apoptosis.
Yeah, that's a very excellent point. I wish Sam was able to jump in here like we had hoped, but that's okay. Whenever he comes back, we can always come back to the slide.
He's in the side chat right now, and I'm not sure if you can see it. He's typing. He says that TP53 is considered the guardian of the genome and is the most mutated gene in cancer.
And we'll definitely get to P53 at some point further down, but I wanted to go a little bit more in depth into differentiating proto-oncogenes and oncogenes. Did Sam have anything else for right now about tumor suppressors or proto-oncogenes?
The last thing he put is that when it is mutated, you see lots of rearrangements, deletions, and copies. And I think we have a question in the live chat as well. I'm on my iPad and the computer. With the likelihood of a cancer cell to mutate, could they mutate to trigger cell death or revert back to a, properly functional gene?
Would you mind repeating that question for me one time?
Yeah, the question from Mike Duffy says, with the likelihood of a cancer cell to mutate, could they mutate to trigger cell death or revert back to basically a regular cell?
Well, the answer is yes and no for that. Actually, most of the mutations that occur, 95 to 99% of them, those cells get either identified by the immune system and removed from the body, or they go on to develop so many deleterious mutations that the cell can no longer function, that the cell can no longer live, and those lines die off. So that happens to the vast majority. That mutated cells will most likely, if you were to place a bet in Vegas on any one mutated cell, 95, 99% of them are going to die off. They're not going to become cancer. But the problem isn't that most of them are killed off. The problem is that there's so many cells and we live for so long that even if there's like a 1% chance, the likelihood of getting it is over a lifetime, over decades, is significant. It's not so close to zero that you can be ignored. So it's likely that those kind of cells die off more often than not. But in response to Mike's question, it's very unlikely that they're going to get that one mutation and then just so happen to get the one mutation that reverts them back into a healthy cell. It's not very likely. I guess hypothetically it could happen, but it wouldn't be anything I would be hoping for if I had a cancer or something like that. When you're at the point of having a tumor or cancer, you're beyond the point for a single mutation to really have that kind of effect. Is it possible that one cell receives one mutation and then at some point it receives a mutation that cancels out the cancer proning effect? It's possible, but I don't think it's anything worth spending a lot of time on because it's such a hypothetical thing and it really doesn't add much to the discussion. If that makes sense. But a very nice question from Mike. Was there anything else, Drag, or did Sam communicate anymore?
He can talk in a minute. Let me check the chat 1 second. No, yeah, you're good to go.
Okay. So just to dive in a little bit more about proto-oncogenes and oncogenes, because this is a very complicated subject. I know when I was coming through my biology courses, I have a BA in biology from the University of Texas. This one concept was probably one of the most tricky things for people to appreciate in our molecular classes. So once again, proto-oncogenes are normal cell genes. They are heavily involved in the cycles of growth, division, and healthy cells. They maintain that balance for cells, when to divide, when to grow, when not to grow, when not to divide. These are non-mutated genes. An oncogene is a mutated form of a proto-oncogene. It's likely that the oncogene is an overactive or overexpressed form of a proto-oncogene. In genetics, this is usually called a gain of function because the normal function is notched up, is amplified. And because it's amplified, there's a gain for the cell. The cell has gained a function it normally wouldn't have. It's in a state of hyper cell division, hyper growth in response to the oncogene. So here's a nice slide that kind of shows how this process can be visualized. You'll see that we're looking at a chromosome at the top, the DNA, and then the specifically blocked out is the sequence of a proto-oncogene. Some type of translocation or transposition, which is basically movement in the DNA, movement from one location to another location, can occur. And you get more of these stimulating proteins that cause growth, that cause division. Sometimes you can get multiple copies of one gene put in that will do this, have the same ending effect, but not necessarily the same mechanism. And at the very end, this is actually probably more likely, is that you get one small area that is changed, and that one small change can lead to a product that either has a larger response in the cell, that is more resistant to degradation, that is just functionally a little bit more unique, that doesn't fall or fit in the normal pattern that that growth factor would have. So when we take this back into how cells develop cancer, we'll take a cell at stage 1. It acquires some sort of negative cell cycle regulation mechanism, whether it's some damage in a tumor suppressor, in DNA repair, even in what we were talking about before. It can be any of those type of things that cause the cell to begin to hyper-regulate its division, its growth. And this is a slow process. I know these pictures kind of tend to make it look like a nice, neat package, but we really have to include time as a big factor. We're thinking about cells that are the great, 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 great granddaughters of cells that acquired the original mutation before the next one comes into play. So one of these overactive cycles is upregulated, whether it's a growth factor or what have you. And you start to accumulate a second one, and the cycle ticks up slowly, one by one, until you acquire a certain level or a certain balance of mutations that throw you over into that category of becoming a cancer cell that mutates rapidly, that accumulates tumor growth factors rapidly, that follows with all the things we've been talking about, the excess of the growth factors, the excess of loss of function of the cell cycle regulators, the addition of ways to evade the immune system, and all those things have to combine together like a nice little recipe in a certain way that produces the end result of a tumor or cancer. And I think just to reinforce this image kind of shows the timeline I've been talking about. You can see that we're talking about decades where you will get a very small group of cells that it's not noticeable in the body. And over a decade, it will become maybe the size of a centimeter or something. And eventually, it will get into that phase where it's become cancerous, where it has the ability to spread or to metastasize. And at this point, the cells will grow and divide very rapidly, where we're talking about months. Or in a month, in 30 days, somewhere, you will notice a lump in that new area. This is the point where you have metastasis. This is the point where cancer becomes very deadly because it's spreading, and that spread is unchecked if it's untreated. So we're about to talk about epigenetics, but I wanted to know if I've been clear so far, if there were any questions, if there was anything else I could clear up to this point. And I'm wondering if Sam has come back.
Yeah, I think everybody's quite clear. I think in the live chat, I've been answering questions as we go along in there.
That's a great idea.
Everything sounds good.
Sam, did you want to add anything about proto-oncogenes, tumor suppressor genes, DNA repair? I saved the DNA repair because we're going to talk about BRCA later, and I saved the tumor suppressor because we're going to talk about P53 later. But if you want to add something now, I'm more than happy to go back a little bit.
I think that, DNA repair genes tend to be, because TP53 is technically a DNA repair gene.
Mm-hmm.
It's considered the guardian of the genome, is the phrase I've heard used in cancer genetic circles. What I will say is that what you have is you have things that stop cancer, that get inactivated. So it's like the brake lines of your car being cut, and then you have things that are almost like pushing down on the gas pedal, and that's an oncogene. So something like, you'll have a growth factor receptor, and usually what that has is you have a growth factor, and the cell's able to grab it. And when the cell grabs it, it then has the signal to divide. And instead, with the mutation, it'll seem like the cell is constantly grabbing a growth factor, and it just continually divides and divides and divides in that cancerous way. Whereas if the cell doesn't have any way of repairing itself or dying when it's irregular, it can continue to accrue damage until it becomes that cancerous lesion. And that's all I wanted to sort of clarify. I mean, you hit upon that, but just nailing that down in detail, because most mutations to cancer genes, and most cancer genes, I believe, are actually tumor suppressor genes. Oncogenes are a little bit rarer, and that's just by virtue of the fact that when you have most mutations are deleterious mutations, and to have something that turns on cancer in the way that an oncogene does, you need almost another functional activating mutation, which are rarer. And so it's just percentage-wise, and in terms of the process being a little bit easier for the cell to overcome. that you get more tumor suppressor mutations. But that was just the last time that I had checked. I don't know if they've done more analyses, but that's my understanding. And I can clarify any of that because I realize there's a lot of terminology there.
And I know, I think I'm 100% with you on that. I think you said that very well. So I appreciate you maybe bringing it back home. I know I've been really repetitive, but I'm doing that in the hopes that it makes things a little easier to break down, because for people who haven't been exposed to this very much or very frequently, it's a lot to take in all at once.
Yeah, it's hard concepts, and we should also say is that when we're talking, like you said, there's no two cancers alike. And I had my experience when I did a little bit of cancer biology is that cancer is like a case on the edge. It's a group of, every case is a case on the edge. So it's incredibly hard to subtype cancers on a molecular level any better than we can just do it by doing the histology, like looking at the cells and the cell type from which it originated. And it's an incredible thing where it's just super hard to understand biologically what the cell is doing during cancer. It's a really horrible and messy process, and that's kind of that's kind of one of the biggest things that you have to understand about cancer genetics is that this is something that's really, really hard, and there's millions and billions of dollars being put towards it for that reason, because it's really hard to understand what exactly is causing the cancer. It's that causation versus correlation that you mentioned earlier.
Yes, I mean, there are newer and newer techniques coming out for studying this stuff. They're starting to finally be able to colonize just single lines of mutated cancers so they can examine them more in depth. The amount of examination you can do from things like biopsies, from things like examining how people with cancer live, is very limited in terms of defining the molecular mechanisms that are in play. And there are methods that are coming out that hopefully will give us better answers to those kind of questions and what kind of those mechanisms we can take advantage of in terms of treating people with cancers. So yes, you're very right on the fact that more research is needed, and by far it is a concept that is the horizon is nowhere in sight, but we hopefully are inching our way closer and closer to get that goal within our grasp at some point. So were there any other questions in the chat?
Yeah, I think Saunders is kind of confused as to what point mutation is, and I think he was asking if the point mutation was causing an oncogene. I think he just has kind of a mixed. He's kind of confused with some of the terms. That's all.
No, I'm glad you brought that up. So a point mutation is a single base pair mutation in a gene, in the genome, at some point in the DNA. So if you can take a look on the right where it says a point mutation in the gene, in this instance, it's referring to the case where one of the base pairs in the gene for that proto-oncogene has been altered. The alteration in that one base pair, that one letter of the DNA sequence, can cause a different type of amino acid to be inserted in that protein. And the effect of one gene can be vast in terms of how the protein functions. You see that protein that's formed underneath the point mutation gene is a different color than the rest. That's supposed to indicate that it has some unique property. In some instances, that product can be more stimulative to the receptors. It can cause a faster rate of stimulation, a stronger response for the receptor. In other instances, instead of being broken down in the normal rate, that signal can last much longer. And in both those ways, you're ending up with a longer or a stronger signal that encourages the cell towards growth and division at a rate that isn't the normally prescribed rate. That isn't what the cell's initial function was supposed to be. I hope that kind of clears it up for him. Is there something else you want to add to that, Drag?
No, I think you actually cleared it up for him. But let me double check in the chat real quick to see if he's on the same page. Okay, yeah, he says, "Oh, I see." All right, cool. Yeah, folks. Let me add a little bit just as an oversight. My dear child, calm down. Don't feel bad about asking questions because we are going over a lot of stuff. But take this into consideration that a lot of the stuff that we're talking about would be taught over, oh God, a course of 3 or 4 weeks at least. And so you're getting kind of bombarded with a lot of terms you're not familiar with, and it's okay to say, you know what, I'm a bit confused as to how this becomes this. So don't feel bad about asking us, and it's not a stupid question if you're genuinely kind of confused or lost. But let us know, because all the stuff that we're talking about here builds on one another. And that's part of the reason why people kind of have a hard time getting into biology, because if you don't understand these foundational, these kind of foundational topics that we're going over, it makes everything afterwards like incredibly hard to understand, almost impossible.
Excellent point, Drag. Mm-hmm. Yes. Again, Drag, I think, is breaking it down even further. I mean, for me, these are things I'm still working on grasping, and my education has been well over a decade in terms of medicine and biology. And it's not anything that I expect to ever master one day, because it's rapidly changing, but because it is so dense, it is so complex. And I rely on other people to explain things, to talk me through parts that I don't understand well enough to explain to patients.
Could I also point out that, I mean, when I went through this learning curve, it isn't easy. And I had to eventually get people, I had to say to them, look, stop telling me about it. Just draw me a picture. I could not grasp some of this stuff. I had to have pictures drawn. And then when the penny drops, you can then progress on further. So if anybody's got anything that they're not understanding, type it in the live chat. Go for it.
Yeah, and I love PowerPoints. I love being able to go to pictures and show things. And maybe I should be using my mouse to kind of point to things here and there. I can try doing that in some of the future diagrams. But for me personally, a lot of my education came in learning these things about genetics from watching videos, because there are a lot of really good videos put out by NIH, by Oxford University, by different places that show how these things happen. Not in real time, but in a time frame that we can appreciate and maybe grasp a little bit. So I would have included things like that if it was appropriate, but I don't want channels like Fiona's to get flagged for showing other videos and things like that. So if you guys have questions afterward, I'm happy to point you to videos that show these things happening, not in real time, but in a way that helps you break it down. That definitely helped me break it down in my education. So if there's nothing else, let's move on to epigenetics quickly. Not that it's an easy topic, but I think a lot of the confusion with people in the way they use the term epigenetics is from the original definition. So this definition stretches all the way back to World War II. And I can't think of many terms that applied in World War II that are still in use today. But this term was created by a doctor named Waddington. He defined epigenetics as the causal interactions between genes and their products which bring the phenotype into being. That is not the definition we use anymore. This definition is just for the sake of being able to examine where we've come to now. Currently, we understand epigenetics as the study of heritable changes in gene expression that occur outside, that are independent of changes to the DNA sequence. So what does that mean? If you're not changing the DNA, how are you changing the expression of genes? Well, it turns out there are a lot of things beyond the genome, beyond the sequence of A, T, G, C, that can affect how often, and if at all, things are made into products, made into proteins. Specifically, some of the ones that are mentioned in textbooks are DNA methylation and histone modification. So DNA methylation it's a modification that's made to DNA in cells. You add what's called the methyl group, which is a carbon with 3 hydrogens attached to it, to a point on the DNA base, or not the base, but the DNA backbone. And this doesn't signal anything, but if you get a lot of DNA methylation, it can actually reduce the amount of times you get products from that gene. So this is a way outside of affecting the direct sequence of the DNA that you can think of it as a dial. You can either turn down the amount of the gene product by methylating it, or if you remove the methyl groups, you can actually increase the amount of gene products that come from that gene or that area of the chromosome. Histones are...
When I first... Oh,
go ahead.
I was just going to say, when I first heard about this, when I was taking genetics, I had to think of something somewhat, I guess, clever for me, or an illustration for me to remember. So I thought of those, I don't know, those menus that you see outside of food trucks, and how people will occasionally write on them and change the price. And so they can change the price, but everything on the menu stays the same. And that, to me, I guess, was somewhat analogous to kind of, it's not a perfect analogy, but at least it gave me a general idea of what was happening.
I like to think that the genes are a pantry, but the epigenetic state is the recipe for what you're making. Well, so you're using, X amount of gene A and then Y amount of gene B, et cetera, et cetera. And it's just, epigenetics, as I understand it, kind of the area of my research, at least part of it, is this could be how the DNA is packaged. What regions of the DNA are open such that those regions can be transcribed and then expressed in protein. It can be, the dimensional structure can, help facilitate little microenvironment, increase the that can.
Is Sam breaking up for anyone else?
What?
Yeah, he's breaking up for me.
Yeah, he's breaking up for me as well.
Sam, yeah, I'm not understanding what you're saying. Would you mind refreshing and coming back in and picking up where you kind of started?
Type in the side.
Reconnecting to the Wi-Fi.
Ground control to Sam. Are you there? Do you read us?
Apparently not. Houston, we have a problem.
Yes.
I can hear you. I'm hearing you loud and clear.
Oh, you're breaking up for us really badly.
Yeah, I can hear you. That's not the issue. The issue.
Okay, you sound good now.
I can hear you or not. Okay, good. Well, my phone is iffy with the Wi-Fi, I think. But yeah, what part did you miss? All of it?
Start over. Why not?
I mean, the way that I understand epigenetics is that it's basically any change to cellular state that is heritable that is not genetic. Sort of a layer above genetics. So this could be something like the three-dimensional structure of the genome, which impacts how much genes are expressed. This can be what portions of the genome are accessible, how tightly they're compacted and how tightly they're not. This can be modifications to the genome or to the packaging. And this can even be stuff like proteins that are floating around, protein aggregates that are floating around in the cell that are inherited upon cell division. So it's a pretty broad field, and I'll say as a researcher in the field that we have some idea of what's going on, but not a great idea, at least not yet. Not to the extent where we have an epigenetic code that is as robust as the genetic code.
No, I think you hit the nail right on the head with that. Epigenetics, while we appreciate that it is involved in tumor formation, we cannot quantify, we can't appropriately describe how much of the variants, how much of the variety of cancers we observe are due to epigenetic changes. And so before we got in that whole cycle, I was going to just briefly go through histones. Sam mentioned it really quickly, how tightly the DNA is wound, proteins on which the DNA is wound up to make that really dense structure. when you see the chromosomes in the slides that are really nice and easy to see, that are very distinct from one another. Histones are the proteins that do that. And like Sam said, if you wind up DNA really tight, it's not as easy for the appropriate molecules to get in there and perform their action to make the proteins, to read and express the DNA. And so that's a factor that can and does change the amount of DNA expression that occurs from an area based on how tightly it's wound, based on the histones that do the winding. So again, just to reemphasize, epigenetic chains, we acknowledge they have some function. We can't particularly quantitate and say to what extent it is involved. It doesn't appear to be a main player in terms of being a Rosetta Stone in understanding cancer. We do appreciate that it is there. It does have a role. So I got a nice slide now to illustrate a little bit of this in a very brief way. So you can see normal cells, epigenetic changes are occurring. These aren't mutations. Again, we're not talking about DNA becoming altered at this point. But epigenetic changes can happen. There can be accumulation of, for some reason, inhibit areas of genes from being expressed. There can be tighter winding around histones. There can be increased methylation or other varieties of epigenetic changes that occur to cells. And we have 3 examples here in this chart. You can see in the top 2, going from left to right, some epigenetic change happens and you have a cell that is now primed, or that is a little more likely to undergo transformation into cancer. In the top example, you first get the epigenetic change, and then you get a DNA change further down the road that initiates the cancer. In the second row that goes right through the middle, we see an epigenetic change that is directly linked to the cancer that accumulates over time to the point that you have the prerequisite factors to cause the cancer. And then, of course, on the bottom, you have your normal mechanism that we've talked about at length, that DNA alteration is the main mechanism for cancer. And that bottom one, that is where most of our cancers we hypothesize are coming from. And we're going to talk about how that comes up in a little bit. So that's really where I wanted to conclude with epigenetics, because we can't appreciate its full role at this time. So now that we're done with that, we'll start talking about hypotheses behind development of cancer. Were there any questions to this point that you guys have picked up in the chat?
Exiled asked a question, asking, could a cancer patient be given histones? I've said I don't think so.
No, it doesn't quite work like that. If you think about it, the body can only, especially if you take the protein orally, your body and the acid and everything, and it breaks down histones. So your body wouldn't be getting histones. Even if you were to inject histones right into the blood, histones are too large. There's not necessarily a port in the cell membrane to transport those histones through. They're probably too big to be transported directly into the cell to bind up the DNA and make it less likely to be expressed. And plus, it would be like scattering a bunch of pellets on the ground to try to hit one spot where you're trying to actually hit. It wouldn't be a very effective therapy, in my opinion, just because you would be affecting every gene if you tried to do something like that. And I'm not sure to what extent it would actually work.
Yeah, the histones wouldn't know where to go.
Yeah, exactly. They don't necessarily have labels that say, hey, histone, you go here. They would just kind of scatter randomly, I would think.
Yeah.
But an interesting thought. I don't think anything like that has been tested. But for reasons I think that are kind of obvious, it's not highly thought of or regarded as a primary mean to solve this problem.
Mm-hmm.
But a good thought, nonetheless. Okay, so any other questions?
No, I think that's it. I think we're up to date on our questions.
Good, good. So let's talk about the cancer stem cell theory or hypothesis. Forget I said theory. All right, so a lot of recent work has gone on to suggest that cancer changes are caused by dysregulation of any one of hundreds of genes, like we've talked about, that lead to the formation of a tumor. This is also called tumorigenesis. The mechanisms that this can occur by are not necessarily 100% understood, but in this hypothesis, they bring forward the idea that you have a primary tumor, and in this primary tumor, there are cells with lots of these mutations. They're not all unique, but there's different categories, and you can see they've divided up the categories in color. The yellow cells are part of the tumor that have a certain type of change. The blue ones have a different type of change from the green, from the orange. These cells will be very susceptible to chemotherapy just because of the changes they have. But one or a few of those cells in there are going to be resistant to the chemotherapy. So when a person gets that chemotherapy, the other cells are going to be killed off, but that red cell is going to survive. And once you've eliminated its neighbors as competition, after the chemotherapy, that cancer comes back stronger, because now the only parts of that tumor, only parts of that cancer that have gone on to survive are already resistant to chemotherapy. It's almost like a synonymous theory to antibiotic resistance. Like when you give antibiotics to a patient to kill the bacteria, well, you kill all the bacteria, but any bacteria that's resistant to that antibiotic you're giving, they're going to take over the show. And that's kind of what this hypothesis brings to the table. It says, hey, we're treating people for these cancers, and we know that chemotherapy only cures 99.99% of the cancer cells. Well, that 0.001%, when you think of a ball of cells like in our primary tumor here as being 100 billion cells, a trillion cells, something like that, you're missing out 100,000 cells that are now chemotherapy resistant, that are going to go on and invade and be unopposed by other cancer cells occupying space, taking up resources from them. You've kind of unleashed them on the body. So this is a hypothesis. Again, I want to repeat. This is a thought process that has been brought forward. It's questionable how much of this actually applies, and it's likely that it only applies to some types of cancers. We don't have a way to detect this yet. We don't have a way to demonstrate the validity of this hypothesis, so it remains a hypothesis to this point. But specifically, they think that cancers like brain cancer, leukemia, colon cancer, and breast cancer tend to follow this pattern where chemotherapy is given, and then you'll see later down the road, these cancers come back stronger. I don't know if everyone's aware of John McCain's brain cancer, but it kind of fits this picture. He had it treated, I think, or he had colon cancer or some kind of cancer that was treated with chemotherapy in the past, and now it's back and it's spread and it's stronger and it's resistant to the chemotherapy. Very good, unfortunately, because of that. That kind of fits in with this hypothesis.
Did you say John McCain?
John McCain, yes.
I thought his was a primary in the brain, a glioblastoma.
Well, maybe it was.
I think it was a glioblastoma.
Maybe I'm thinking of someone else. I might have my celebrity figures mixed up.
I'm sure John McCain's is primary tumor.
Okay, well,
but that's only because I'm rather fascinated by glioblastomas.
You might be right. I might have been thinking of who was the. Well, never mind. The important thing is cancers that fall in that type of pattern, even though that's not the. I'm not accusing.
Sorry about that.
John McCain of having that cancer, but thank you for correcting me. That type of pattern fits very well, and we do observe that happening for some patients. Unfortunately, the prognosis is very, very poor with that type of picture. So, were there any questions about that hypothesis? Because that's a pretty tricky one. It does not apply to most cancers, but in probably a subset of cancers, it seems to really explain the picture quite well.
I don't think there's any questions. There's none that I can actually see. I think one of the reasons why, people will have grasped that slide quite easily, is because it mimics what we describe happening in evolution.
You're right. It kind of does.
Yeah.
the strong get stronger and they take over,
Yeah.
kind of thing. But it kind of stems from that. But if you read the paper that's quoted at the bottom, you know,
Mm-hmm.
they very much, I think, got that idea from microbial resistance, which is obviously
Yeah.
linked to evolution.
Yeah.
So I'm glad that everyone's following quite well.
They
So
are.
there are a couple other hypotheses as well. They're also very difficult. They require a lot of background knowledge to understand. But we do know that in several cancers, such as cervical cancer, that viral infections, such as HPV, greatly increase your risk for developing these cancers. So according to this hypothesis, it's called in some instances the limited resource hypothesis or the antivirus hypothesis, that a tumor cell would be the only type of cell that is able to respond to viral replication. So in the antivirus section, we assume that in your body there's always one or two cells that are slightly closer to that tumor phase. They're at a higher growth rate than other ones. They're a little more prone to becoming tumors or cancers. And you'll get a virus that attacks the body, that enters the cell. And it can either occur through one of two mechanisms. On the bottom, you'll see the other cells in the body get attacked. They go through the mechanisms that slow down their cell growth to respond to the viral infection, to respond to any DNA damage the virus tries to do. But the cell that does not have those check marks, that has the whatever mechanism to grow without check faster, is going to take over, again, similar to the previous hypothesis. It's going to grow with less competition because the virus is inducing a state of low growth in normal healthy cells. So basically, in that thought, the virus is causing the mutated cell to outcompete the other cells because it's giving that cell an advantage. It doesn't respond to the viral infection. Whereas the normal cells do get suppressed, they do stop replicating, and that leads to cancer. In the top example, the limited resource example, you see that a virus directly causes tumor suppression, or suppression of tumor suppressors. And that leads to increased viral replication that increases your risk for developing cancer. So in other words, it could be thought of as a mechanism where the normal cells are unable to respond to the signals that the virus send out. This causes biological changes that ultimately lead to some sort of response. The body tries to get cells to grow and replicate in order to overcome a deficit that's caused by viral damage. And again, any type of cell that has a small change that lets it grow a little bit faster than its neighbors is going to respond that much more to that kind of a signal. So let's take a hypothetical. Let's say that someone has hepatitis. Hepatitis is a viral infection of the liver. It's a chronic infection. That virus is there. It's damaging the liver constantly. Well, in response to that damage, the liver is going to send out signals to the cells that, hey, grow, divide, and try to overcome the damage that's being done so that the way the liver can maintain normal function. Well, now you may have one or two cells or a handful of cells in the liver that have had some sort of mutation or some sort of change that have led them on a tumor-forming or cancer-forming path. They are much more sensitive to that grow, that divide signal, and they will gladly take that signal and ramp up the process in which the cancer develops. So it would make the cancer appear much more quicker in a time frame that it's not as likely for your body to be able to fend it off through its natural immune ways, natural immune processes. And those are the two mechanisms by which viruses are thought to possibly contribute to cancer development, such like in liver cancer, cervical cancer, and things like that. And I know this picture isn't as nicely explained, but this is the best one I could find. So I would love to hear some questions about this. I'm not sure I explained this very well.
Well, I think I
And
got
this slide is very confusing.
when you were talking about suppressing the tumor suppressor.
Mm-hmm.
And I thought to myself, the brakes on a car suppress the speed. But if I cut through the brake pipe, then I've suppressed the speed suppressor. Therefore, going down a hill, I'm going to get faster.
That is the best analogy I think I could give, yes.
Cutting the brake pipes.
But yes, maybe the virus is just going in there and cutting the brakes on some of these cells, and they go haywire.
Mhm.
And the other hypothesis is it kind of a combination of a virus with the previous stem cell hypothesis, that hey, maybe the virus is actually suppressing all the normal cells. Meanwhile, the cell that is precancerous, that's pre-tumor, is now unopposed. It can get all the nutrients and all the growth factors it needs because all the other cells are ignoring those factors. So it's an interesting topic. I am not sure how this would be tested. I'm not sure how this would be validated. I'm going to let other people come up with that information before I start to really consider this and make this something I would show to patients that teach people who have cancer. This is just FYI for all our knowledge here because we're examining this more in depth. So if there are any other questions on either of these two hypotheses, please, let's talk about it. I would love to have a little more guidance in terms of how to educate people on this, because this is actually something I've never explained to anyone before.
Well, we have got Exiled in the live chat who's saying you've explained it very well. So there you go.
I was hoping for some more questions, but that's okay.
Yeah. Well, we've got a question. By the time those cells become suppressed, is it too late to shut down and die naturally? Or
So we're talking about the virus infecting the cell and causing the suppression of the targets, suppression of the suppressor genes.
Yeah. So
it's not too late to kill the cells. The problem is there is no mechanism to identify these cells. In terms of viruses are so small, you can't look at the cells in a microscope and identify, okay, this one's been infected and this one hasn't. The same thing for the body. Unless the cell starts to express parts of the virus on the surface, the immune system has no way to identify that cell's infected.
Yeah.
That is the normal mechanism. So when your body gets infected with something like the flu, don't get killed by the flu, but break parts of the flu up and start putting them on their surface. So that way the immune system can pick them up, identify them, hey, this isn't part of normal cells, the normal cell membrane, and start to find and basically chew out the cells that have that viral defect in them. Mm-hmm. And that's how your body fights viral illnesses. That's why you feel crummy when you have the flu, when you have any kind of virus, is because your body is literally almost performing chemotherapy on itself in a very loose sense. The immune system is chopping out cells from your own body that's detected the virus in. And in this instance, that mechanism isn't in play yet, because that takes time. If you're familiar with viral infections and immunology, usually you get an infection and 2 or 3 weeks later you get the symptoms. It takes almost 2 weeks for the symptoms to appear. It takes that much time for the cells that do get infected and live on to chop up the parts of the virus and put them in their cell membrane. So that way the immune system can become educated about it. So this system takes a long time. And that's why usually by the time you're having a fever, you're having symptoms, you're not able to pass the virus on for the most part. Your body is already fighting the virus at that point. It's that time, that week beforehand, that's when you were passing the virus on, when you weren't symptomatic. That's one of the reasons why we tell everyone, wash your hands, don't put your hands near your mouth, cough into your shoulder, and all those kind of things. You never know when you're technically infectious. So I hope that kind of clears it up, because the immune system won't be able to pick up this point in the diagram where the virus goes in and turns off the tumor suppressor genes. But you can't appreciate that once that happens, you are at greatly increased risk for developing excessive growth, excessive replication. And in the cells that don't normally get killed by the virus, like here, they will continue to have that tumor suppressor gene deactivated, and they will continue to pass that on to their daughter cells. It was a great question.
Yep, we've got another person saying that the explanations are excellent.
Well, thank you.
Yep, that's is it Saunders or Saunders? I'm not quite sure how to pronounce his name.
Well, thank you, Saunders. Colonel Saunders. All right, so we'll go on to the next hypothesis then, unless someone wants to interject something, which I'm happy to entertain, of course.
Somebody's asking who's just arrived, who's presenting. The person presenting is RSC, who is an MD, and he is presenting his knowledge on this topic. So there you go.
Okay. Well, let's talk about another hypothesis. This is like the molecular basis of cancer. A lot of it's going to be very repetitive of what we've talked about before. We talked about carcinogens, about things like radiation. But in this model of cancer, they also include viruses. Basically anything that can cause damage to the cell, damage to the DNA. And I think in this cycle, you'll actually appreciate a little bit better how things happen. So any of these consequences from up here can damage your DNA in the nucleus, in which case your DNA is damaged. But there are so many mechanisms in the cell to repair that damage that most of the time, we're talking 90+% of the time, repair happens and you go back to being a normal cell. And that cell goes on and has daughter cells, and there's really no consequence for the patient, for whatever organ or tissue that's involved. Other times, you get that DNA repair is unsuccessful. Very inoften, you get the mutated cell. More often than not, the cell is going to be programmed, it's going to die. And that's not pictured here. Or the immune system is going to detect there's damage, and the cell is going to be selected for degradation, for apoptosis. So this is a very rare instance, but remember, like I said, we have trillions upon trillions of cells. We live for decades upon decades. It's just a unfortunate fact of the number of times that our cells are going to undergo insults, that eventually one of them is going to slip onto this mutated portion. And if one of those genes affects something, like maybe it prevents cells from undergoing apoptosis, well, now that cell line isn't going to commit that cell suicide if it gets another mutation. And another mutation can acquire further down the road. Like we saw several slides ago, this is likely something that's going to take a decade before another mutation just happens to hit that 1 in a billion cells in that small area that has this already given mutation. In which case, you might have an oncogene activated. A tumor suppressor might be cut off. That brake might be cut off. Or even something like a growth factor. It may become upregulated, in which case this cell now has become either a tumor cell or a cancer cell if it's malignant. And it gets a massive growth, a rapid growth, a progression of many cells over a short time. But this little fact down here is that cancer cells don't grow faster than normal cells. They just grow uncontrolled. The time it takes for a cell to grow, divide, it's a set time. There is a speed limit to how fast that can happen. Cancer cells don't go faster than that. They just go from division to division continuously without any time, with much smaller time in between the divisions. And that's what we mean when we're saying that their growth is uncontrolled. They don't stop to pause. They have a baby, and then they spit that baby out, and then they're ready to have the next one immediately. So again, we have another picture showing the same pattern we've talked about. And in this case, it shows, you can see we have methylated and unmethylated DNA bases exemplified. You can see a very complicated pattern in which, identified here, it's a change. This change is, it's called a stochastic differentiation in the methyl group. That just means there's a different physical pattern, a physical shape. So you decrease the methylation in this area because these have become unmethylated. The blue ones have the methyl group. Methylation tends to decrease the amount of products you make from that gene. So once you remove those methylation products, you start to get more expression of this area. And if this just happens to be the right area, you can get increased growth, increased divisions. And eventually this one cell can become a large colony of multiple cells that have this pre-cancerous condition. And should they go on to later develop further mutations, further changes, they can go on to become cancerous. And it's a little nice picture here of how that's done. So I think this is actually the last hypothesis we'll talk about today. This is the Knudson's two-hit hypothesis. This was a very popular hypothesis when I was in undergraduate. I don't know if it's still as popular now. Sam, do you have any idea whether this is still heavily taught and heavily mentioned, or Drag? Are you hearing a lot about this in your classes?
Can you repeat that?
Are you the two-hit hypothesis? Are you familiar with that?
No. I think that's something we're going to go over next semester.
Ah, I see. Okay. So a little pre-knowledge for Drag in his future coursework. The two-hit hypothesis. It's almost exactly as it sounds. Basically, the hypothesis was that you need two instances to happen in order to get cancer or to get the formation of a tumor. And it's always been thought of that, okay, you know, one of them has to be something like a tumor suppressor, and then one of them has to be a proto-oncogene, or one of them has to be a DNA repair mechanism, and one of them has to be a tumor repressor. And that these don't necessarily occur one after another. There are two instances. You can either start off as having normal DNA, normal chromosomes, and at some point down the road, you acquire one mutation in one of those areas. Remember, we had the three drivers that I mentioned previously. And then after you acquire the first, a little further down the road, you acquire that second one. And that's that second one that tips the scale in favor of transforming from just a slight overgrowth or a slight precancerous state, pre-tumor state, into becoming that full-on tumor, that full-on cancer. The other cause is you inherit, you're born with one of those defects. You have a loss in a gene for a tumor suppressor as a child. You have a loss of DNA repair as a child. You're born that way. A deficit you'll never be able to overcome, unfortunately, unless we have new technologies or therapies that come up in the future, which hopefully we do. But then you only need one additional hit to form that two-hit hypothesis. And at some point down the road, you get that second hit. And now you have a defective DNA repair mechanism. And now you have a defective tumor suppressor gene. And now you've tipped over into forming that tumor. And that's really the basis behind the two-hit hypothesis. I think this is probably the easiest of the hypotheses to understand. Were there any questions about the four hypotheses we talked about in terms of genes and cancers?
No, everybody's following along great.
Excellent. I mean, does anyone want to add anything?
Well, your next slide was...
this was where I wanted you to take over, Fiona, if you don't mind.
Oh boy. Right, well, retinoblastoma. This is it's a horrible condition. And if you go back to your previous slide, okay, you have your paired chromosomes, an allele on each one. Some children are born with a defect, so they only have one working copy. And these children are at very high risk of a horrible type of cancer called retinoblastoma, where a tumour forms in the retina inside the eye. They only have one functional copy. So they only need one hit and they will form that tumour. Whereas everybody else, they have two copies. And if one copy gets hit, the other copy can take over and a tumour won't form. And for someone without this inherited defect, for them to develop the retinoblastoma, they would have to have those two hits. But this is a cancer that it tends to pop up in childhood because you only need this single event for it to begin. And there's that lovely little picture that shows you what goes on. And if you want to explain your picture.
So yeah, this is exactly what Fiona was saying. That in this picture you see you have an egg and a sperm. And in this instance, we're going to assume that the sperm has the defective copy of the gene. So when you form the embryo and eventually the baby's delivered, the baby has one good copy and one bad copy of the gene. And they come from, the inherited. At some point in the life, because they're susceptible to getting only one allele knocked out, some portion of the eye happens to get that damage, where the good portion of the retinoblastoma gene becomes inactivated for some reason. And when that happens, they go on and they fall into that phase where they begin to develop a tumor. They develop the cancer.
And remember, it's only one single cell within the retina that has to have that specific damage happen to it. And then the tumor will start to form. It's not the entire retina. So we're talking about if this happens in a single cell, and given how many cells are there, even although the probability of a hit for a certain, one cell is pretty low, the fact is the huge number of cells that are there in these children, it usually ends up that it happens to one of them, and that one cell will then go on to form the tumour. And all the other cells that have maybe suffered some damage, they don't. It has to be very specific within that gene for this cancer to form.
So you can probably appreciate this picture instantly. When you look at this child's eyes, you notice something is not right just immediately. I think everyone can see that his left eye, the eye on the right, it has what we call the white reflex. Now, this is not a normal reflex, but it's not always indicative of disease. But retinoblastoma is a very known disease state that causes children to have this condition. And like Fiona says, it forms on the retina, which is the back of the eye. It's where the nerve cells are. And this tumor becomes quickly malignant. And it spreads locally. So it spreads within the eye socket, to the bone, to the orbit, to the rest of the area in that area. And unfortunately, because going back is directly to the brain, it is very likely to spread to the brain and spine as well.
Yeah.
And like we talked about in that two-hit hypothesis, the reason they think children get it more often is because they've inherited the one defective copy. And the RB gene, RB1 gene, it creates a protein called retinoblastoma. This is a tumor suppressor gene. It's involved in many cancers, actually, but it was first studied by Dr. Knudsen, the man who came up with the two-hit hypothesis. This is where he got that from studying these patients.
The horrible thing about this cancer is that the only treatment they have for it is to remove the eye. But even worse,
Yes.
The other eye is at the same risk.
Yeah, and because they've inherited the bad copy, that risk never goes away.
Yeah.
So here's a good picture of the eye. You can appreciate the retina is this very thin lining of nerve cells that cover the back of the eye. That sense when the photoreceptors light up in response to light. It lets you produce images that let you see. And then you can see that once a tumor involves just one of those cells, it starts to grow, occupy, and invade all the other cells around it. And you can go on YouTube and see all sorts of pictures. It's a very disease to have to look at.
There's actually
but there's good news.
There's an instance of, I think it was a lady in America sending a photograph of her child to her friend in the UK. And the friend in the UK looked at the photograph and she picked up the retinoblastoma. Just when you were showing that picture, showing how obviously there was something wrong with that eye.
Mm-hmm.
I just thought I'd throw that in there. That somebody just looking at a photograph on the other side of the Atlantic was able to say to that mum, take your child to the doctor.
Yeah.
Just for information. Sorry, on you go.
Well, no, that's an excellent story. Luckily, this is not necessarily a very common cancer. I think I have some statistics here for you if you're interested to hear them. Maybe not. But it's the good news I was talking about is that it's actually one of the most survivable cancers, even though enucleation is the procedure where they remove the eye. It can be very detrimental in terms of, lifelong disability and things, especially if both eyes happen to have the cancer. But the children that get treated early on in the disease, they go on to live full-length lives, which is, a very nice blessing in its own way. Even though, harm has come to them in this form, they go on to live lives. They don't necessarily at greatly increased risk for developing other diseases down the road, even though retinoblastoma, that RB1 gene, is involved in many cancers as well. Here you can actually appreciate where it is on the chromosome. It's highlighted here. It's on chromosome 13. It's on the long arm. You can see it's right here in between the genes for Wilson's disease and then the BRCA2 gene. So we'll be talking about BRCA2 in a little bit as well. So Sam, would you like to talk about P53 or TP53?
Sam's not here.
Oh, he's not here. Okay.
No, he says he'll come in once his phone is charged and he's done with stuff in the lab.
Okay. Well, no, I appreciate Sam's input on these slides and everything. So I'll just go through TP53 or P53. So P53 is a tumor suppressor gene. It has a vast array of functions in the body. And like Sam said, it's nicknamed the guardian of the genome because it's that important. In almost every cancer we know to date, many, many versions of that cancer have mutations in this gene because it is so crucial in terms of protecting the body from cancer, development of mutations. It functions in many ways. It halts the cell cycle. It prevents the growth, the synthesis of DNA in response to DNA damage. It can even start the cell suicide process if the damage isn't overcome, isn't fixed or corrected. And it can also induce what's called senescence, which is basically a permanent retirement state for cells that, hey, okay, P53 can promote you to being fully retired where you never divide again until you expire. And that usually occurs when your telomeres are really short, when cells have telomeres that no longer are able to serve their purpose, that have been cut down through or after repeated cycles of cell division. P53 says, okay, you're done. We don't need you to divide anymore. Your time is up. And hopefully everyone has heard about telomeres in the past. We can always go over that if there are any questions. So here's a good slide. This is just a fraction, and I hope I can emphasize, a fraction of the tasks that P53 is involved in with the cell cycle that we know to date. So
Could I just throw in here?
But I just wanted people to appreciate how involved P53 is inside the cell in terms of regulation. Go ahead.
Could I just say, that diagram that looks like a map of London Underground, which is so complicated. That's for the P53 gene alone. And we know of hundreds of genes which can be involved somehow in the formation of a cancerous cell. So imagine if you overlaid them all. And that brings home why it's been so difficult to find a cure for cancer, because there are so many different causes. Just throwing that in there for information.
No, that is the exact point I want people to take home from this picture. I don't actually expect people to break this down without several years of in-depth molecular biology coursework. But I can show you simply. Here's P53 in this system. P53 responds to gene regulation. It responds to cell signals. It responds to things happening in the outside of the cell. You see, these are embryologic factors, so it has a role there. And then I don't know if you can see down here, but this FAS receptor, this is the death receptor. When the immune system wants to tell a cell to commit apoptosis, this is the receptor it uses. And guess what? It's involved in it. P53 is involved with that as well. So because you see that so many of the systems that the cell relies on to check DNA repair, to check cell growth, to check cell suicide involve P53 and go through P53, it makes sense that it is mutated in a lot of cancers. And I think Sam probably was going to mention the fact that elephants rarely get cancer from what we know, and they have like 17 different copies of P53 in their genome. And they're thinking that's probably why they don't get a lot of cancer in that species. They have such an overabundance of the guardian of the genome that it makes it even less likely. It decreases their risk of cancer millionfold by having so many copies. And it's quite possible that we could use that somehow to our advantage, but the technology isn't available to induce P53 in cancer cells at this time.
So would that be, so you're suggesting that maybe if someone has a tumor in which P53 is mutated in all of the cells, that a gene therapy with a corrected copy of P53 may be possible in the future?
I mean, that's not far-fetched. I mean, if that's what's broken most cancers, why not fix it and see if we fix the cancer, right? It's a very simple thought process, but a lot of the things we do in medicine are very simple thought process. We identify something that we can alter, and we alter it in conditions where we think there may be a benefit.
Cool.
Okay, so this is a very narrow breakdown of P53's use. This is really just in response to DNA damage and maybe some things that happen outside the cell. This doesn't cover all the functions of P53, but I thought it was just a good example of how it interacts. Basically, normal P53 is bound outside the cell by this factor called MDM2. It's basically an inactivating factor. It prevents P53 from doing anything. P53 is relaxing at this point. But once something happens to the cell, either there's damage to the DNA, there's some kind of abnormality, there's not enough oxygen in the cell, well, now it's time for P53 to get up and get to work. At this point, the MDM2 releases the P53, and then it's active. It becomes an active state. In its active state, it does many things all at once. Like we said before, it can be part of the signal for the cell to commit suicide, apoptosis. Also, its primary function is to stop the cell from progressing down the growth, the synthesis, the cell division phase, and to encourage the repair and restart the cell cycle if the repair is done. And basically, if this side of the P53 chain does not get done in a timely manner, the pressure on the cell suicide builds and builds and builds until basically it seesaws and the cell goes down this line. So that's the mechanism by which P53 guards the genome. It forces the cell to either fix itself and then continue down the normal path. Otherwise, p53 is going to initiate that trigger to kill, for cell suicide. And basically, both of these processes together prevent cells from becoming unstable genetically, from acquiring these mutations, these negative effects that lead and make it prone to developing cancer in the future. So again, here's another wonderful slide that shows even more of the effects that p53 has, and how a p53 mutation can cause or lead towards that precancer state, or maybe even tip a cell line into becoming cancer or tumors. So we can see in this picture that there's a mutated p53 from some previous mechanism. Now you get the things like chemicals, carcinogens, free radicals, proto-oncogenes, in combination with DNA damage. And pathways that would normally repair the cell, like this pathway called nuclear excision repair, they're unable to function. You're unable to repair. In response to the DNA damage, p53 would normally step in and help increase this signal for repair that's urgently needed. But because it's mutated, it can't do that. And so the things that are prevented, stopping the cell cycle, that doesn't happen. Cell suicide, that doesn't happen. The cell doesn't become more specialized. It stays in an immature form. There's a continuation for the cell to express hormones and molecules that increase blood vessel growth to feed these cells that will become the tumor. The DNA repair mechanisms are all but turned off at this point. And stresses like oxidative stress and other stresses, like from physical and chemical factors, can continue to mount on the cell and basically snowball this effect of causing more mutations upon more mutations at a rate that's much faster than we talked about in the past, where we go from one mutation and then waiting a decade before the next mutation is inherited, and then waiting another couple of years before it happens. Inactivation of this mechanism allows for a much more rapid onset of mutation after mutation within a cell lineage in this area.
Just to sort of get an analogy going here. So what we described previously is like, you're going down a flight of stairs, stair after stair after stair after stair, to eventually becoming a cancerous cell. But if p53 gets knocked in the head, it's like being at the top of, the ski jump thing. And when p53 gets mutated, somebody's giving you a shove and off you go down the ski slope. And go whee off the end.
You don't have your tumor cell suppressor for the brakes, so you can't stop.
Yeah. So instead of going down the equivalent of going down a flight of stairs, step by step by step by step, at a normal rate, you're accelerating all the way down that hill because p53 is now not doing what it should.
Yes, and then you see out here, mismatch repair is another form of DNA repair that also becomes damaged when p53 is mutated. It's not effective because in order for these repair mechanisms to work, the cell can't be growing. It can't be making new DNA. The cell has to stop doing all those things in order for these mechanisms to be effective. If p53 can't stop the cell cycle, these mechanisms can't work. And even if they can work, now that p53 is not able to work, it's not able to respond to stress in the cell, it's more likely to accumulate mutations and defects in these repair mechanisms down the line. Just like the step-by-step process that Fiona described, it basically gets erased. It's basically a downward trend once you get this gene mutated. So a very adept analogy, Fiona. I guess Sam's still not here at this point, so we'll move on if there aren't any questions.
No, there don't seem to be any.
Damn, I'm doing too good a job.
You are.
You're doing good.
Yep, keep going.
Okay, so now we're going to move on to our final example. BRCA1 and BRCA2. So these are both genes. They are tumor suppressor genes, but they're specifically part of the mechanisms that repair DNA that's damaged. When these genes are inherited and mutated, you have two copies. One copy can usually make up for the defect in the other copy. It's when you get that second hit that you lose complete functionality that the cancers will tend to form. But because you're at such a narrow window, you only need one hit, the risk for developing these breast and ovarian cancers is much more. So I can talk about a little bit that these cancers they account for about a quarter of all the breast cancers that are inherited. They're about 10% of all breast cancers when you look at all breast cancers. And additionally, they account for about 15% of ovarian cancers. And I don't know if most people know, but ovarian cancers, there's no screening test. There's no blood test that we can do. The symptoms are usually very mild until very late in the disease. So it is a very severe cancer. The prognosis is not usually good. You're usually at a late stage of disease when it's detected, because you only will detect it once it spreads somewhere else, and it likes to go to the brain. It likes to go to other places that we've talked about. So in these kind of patients that they have this condition, a lot of physicians like myself will say, if you're a female, if you are done having children, get rid of those ovaries, because they're just basically ticking time bombs.
Now, I do believe that a well-known person who was carrying one of these mutations was Angelina Jolie. She had both of her breasts.
I can't tell you how many times. Yeah, I can't tell you how many conversations we've had with our oncology department, and every time they want to talk about BRCA1 and BRCA2, they just pull up a picture of Angelina Jolie.
Yeah.
Because people are always asking about that.
She made a very courageous decision. She had a double mastectomy, and I believe she had her ovaries removed as well, because she was at such a high risk.
So likely she was carrying the BRCA1 mutation, because that just has an increased risk for both. But I mean, there's no way to tell. We don't have access to her medical records. But yes, that is the most common example. The questions about double mastectomies, prophylactically, and things like that, that is debated, because as a healthcare professional, we rely on evidence. There's not a lot of evidence saying that is beneficial to do this, because not many people have undergone that. So we don't know what the results are. I'm sure she made a very active decision, in combination with the people who are helping her with her medical decisions before undergoing that procedure. Some doctors will advocate that consider it. Some will say, no, don't do it. But the problem is there's no good information at this time to make a decision. You don't have enough information to make a decision on one hand or the other hand. So basically, I guess most people will leave it up to their patients to do and wait till the data comes in before we start giving advice which path to pursue. So that's just an FYI about how things are taken care of.
Yeah, just to say, Angelina, went public with all of this. It was a BRCA1 mutation that she carried.
Oh, she confirmed that?
Yeah.
Okay. So
sorry, I just thrown it in there.
No, it's actually very interesting, and I think people always perk up when they hear Angelina Jolie, so it's not bad, because we're almost at the end. But here's a little demonstration about how BRCA1 and BRCA2 work. Basically, they're involved with a specific type of DNA damage. It's called a double-strand lesion. So you can see up in this picture here, we have a normal piece of DNA, but this piece of DNA has been cut prematurely here. And not just on one strand, but on both strands, clean through. So how does the body handle this? Well, there's two mechanisms by which the body can handle this. One is called homologous double-strand DNA repair. We are not talking about that. That's a very complicated system. I don't even want to go into it right now. Hopefully, Fiona, you agree with me on that. We'll talk about the other repair mechanism. This is called the non-homologous repair mechanism, and this involves these two genes. But basically, the BRCA protein, it is the detector for when this happens. It is the siren that starts to warn the cell that, hey, you have a double-strand break. It detects it. It binds to this area. The BRCA will actually go to this area, bind, and it will call for other molecules to come and assist it. RAD51, which is attached to BRCA, is the response, the assistant that comes to BRCA1's aid once it detects a double-strand break. And together they form a complex that, through a series of DNA cuts and snips, will restore both ends of the DNA and cause the double-strand break to be repaired. It's a lengthy explanation. I would actually advise, if you want to see this in more detail than what I'm telling you here, there are a couple of really good YouTube videos put on by educational groups that go through showing how this carries out. And watching the video once is worth me explaining this to you a million times. So I would recommend, if you want to find out more about that, I can post links or have Fiona post links after we're finished here for that.
Don't expect to learn it all in the first or second or third go. I'll tell you from personal experience, people think I know a whole hell lot about genetics. I don't. And something that I try to put out there is that even though I've taken one genetics course, it taught me how much I don't know about genetics, and that's really it. And so I'm taking molecular genetics next semester to hopefully shore up my understanding, for my future endeavors. So, for those of you who are just trying to learn it and understand how these genes, impact people's lives and decisions, such as Angelina Jolie, there's a little bit of there's a little bit of gray area when it comes to the decisions we make with our lives in respect to the genes that we have inherited. So there's really no big hard and fast. And I'm just trying to echo what RC was saying. Please check with your primary care physicians and don't just watch stuff on YouTube.
Yeah, I mean, the people on YouTube who are giving you advice, they don't know your picture. Your picture is unique, and that applies to everyone. Even if you just have, something as simple as like, I don't know, a pneumonia, or you have a small skin infection. You are a unique set of circumstances, and I personally would never do telephone medicine. I know people have questions for me and things like that. I'm happy to answer those questions, but I do not feel comfortable treating people over the phone, over Skype, over a Hangout call. For that specific reason, I need to see you. I need to meet you. I need to get to understand who you are, both, as a person and as a patient, before I can give you a good recommendation on a treatment plan. I can get, good information about, some of those things over a phone call or over a YouTube video, but I could never get the whole picture. And I would never assume that there's any substitute to that in medicine. So yes, that's one of the reasons I would say that it's always great to get information, but that information has to be integrated, and you need someone who does that responsibly to help you sort through it all. So thank you, Dreg, for bringing that up again.
No problem.
Any questions to this point? All right, let's keep talking about these genes. All right, so more in depth, we can see that people who inherit these mutations are definitely at increased risk for this two-hit hypothesis that we talked about. PARP inhibitors, don't worry about that. That's a specific type of medication that can be given to help reduce the frequency, or it's an adjuvant therapy that can be given to prevent recurrence of these cancers by, preventing loss of the BRCA1 and 2 function. But basically, you can see in this picture that a single strand break in the DNA here can become a double strand break, in which case, when you have both of the active BRCA1 and 2, the cell repairs that, hunky-dory, goes on, normal survival. In this case, we see that BRCA1, 2 is positive minus. This nomenclature, these symbols mean that the plus means that it's normal, the negative means it's abnormal. So the plus on the left indicates BRCA1 is normal. The minus on the right means that we're missing the copy of 2. So we go through the same situation here. Start single strand break, develops into a double strand break, but we only have the BRCA1 is able to identify the break. It calls for its helper, but its helper can't come because the BRCA2 is what it's needed to form the complex to fix the DNA. Without it, you have a tumor cell that is resistant, especially when you're already giving this medicine here. It's now resistant because this medicine is unable to help. If we go down, it's probably difficult to see down here because of the exit and all that. I don't know if I can move this stuff. I can't really move it, but you'll see we have the same instance down here. Single strand break goes to double strand break, but in this case, it's a minus situation, which means neither copy is present. This is so deleterious to the cell that these cells don't survive to become tumors. They die too quick for that. So it's the specific case where you have only one of those missing. So you don't hear about people developing breast cancer who have both copies that are deleted. Because if you were genetically, if your genotype had both of these copies deleted from your genome, you wouldn't survive, unfortunately. And there aren't people walking around without BRCA1 or 2 at least active, as far as I'm aware. I might be wrong, but that's what I understand.
Is that
People don't live with that.
It basically,
I'm sorry?
basically, if you had both copies broken, you wouldn't have survived through. Well, basically,
you wouldn't have been a baby.
Through fetal development, you wouldn't survive through that.
Yes, that's my understanding.
Yeah. So you're talking about
this is someone who has
egg that wouldn't survive. It would not be a successful pregnancy.
Correct. In this instance we're talking about here is someone who had either BRCA1 missing or BRCA2 missing, and then through some other mechanism, they also got that second hit to inactivate the other one. Those people don't go on to develop tumors in those cell lines because of that reason. Those cells die. They can't sustain viability anymore without those. It's a specific case where you're missing one or the other. So it could be plus minus or minus plus. That's what leads you to these resistant tumor cells with BRCA. And that's why they're so deadly. They're resistant to a lot of the medicines we have, and the fact that it occurs on one side, but the other side, the other breast, is also just as likely to develop that cancer later down the line. So here's just a basic demonstration. These are on 2 different genes, or 2 different chromosomes. BRCA1 is on chromosome 17, and you can see this would be a carrier here of the BRCA1. They have one bad copy and one good copy. And then BRCA2 is located on chromosome 13. This person will also be a carrier if they had this picture. One bad copy and one good copy. So that was the end of my presentation, really. I expected to have more questions. I'm sorry if I've gone on for so long. I know it's been kind of tedious, but I hope the pictures really helped explain some of the points I was trying to make and help things be clearer than they would have been without.
Well, I think you didn't get questions because you were so clear. I think that was a fantastic presentation.
Yeah. Go ahead, Sam.
I was just going to say it was very, I'm sorry I had to sort of pop in and pop out, because with the time difference, it's only about 6:30 here. So, I was popping in to get work done, and then my phone was dying. But in general, it was pretty good. I mean, it's hard to say because the basics, there are a lot of basic principles, but what I will say is the really complex stuff is when you sequence tumors, because that's its own game that is so difficult. And it's hard to really stress. It's, if you're having trouble understanding it, I mean, you and the rest of the scientific community, it's not, we're talking about a disease that we really, really are only scratching the surface in our present knowledge. And I can confidently say that because, you still see, it's not as inundated now, the literature, but you still see so many papers on just like these one little case and this one cancer, and it's pretty crazy.
Yeah, you're right, Sam. I appreciate your feedback and all the help you gave, all the details you gave for putting the presentation together. That was really helpful. Fiona, I appreciate you going through the presentation also. And Drag, thank you so much for showing up. I definitely needed the help since I'm not able to multitask and observe the chat as well as I would have liked for the questions and answers. But you guys all gave me a lot of great feedback, a lot of great input for the presentation. So I really wanted to thank you.
You might want to stop sharing your screen now. Ta-da!
Wonderful.
Wow! Do you know, in technical terms, this Hangout worked very well. I wasn't expecting that. I was expecting glitches everywhere. What I'm going to do is I'm going to be taking us off here. But what I wanted to do was one thing that I think has made this Hangout what it was. I mean, apart from RSC's brilliant presentation, was our audience.
Yeah, they were very engaged.
Yes. And I mean, I honestly think that thanks to the audience, that this has gone down as well as it has. And so I've got to thank the audience for that. We didn't get a single trolling comment.
Oh, it's too bad. I would have liked to have dealt with some trolls.
Well, if you really want that, G-Man, just send me a message on Skype. I mean,
oh boy,
no.
I think that's a good note to go off here. If any, you needed a signal.
Yeah, but I mean, honestly, this has been a fantastic hangout. I'm going to have to watch it again myself. I found it absolutely fascinating. It dragged things up from my dim and distant past that made me realize just how much I've forgotten in my lifetime.
Those frightening genetic tests.
Yeah, the thing that scares me about this is, we just went into very big detail on all this stuff, and RSC will back me up on this. We didn't even scratch the surface, which is absolutely crazy on how complex this topic is. It always blows my mind. I learned so much in this presentation, and yet there are so many other details. Like, for instance, what happens when you sequence a tumor? How do you evaluate? Because we talk about tumors as these big blocks, but in reality, the tumor doesn't stop mutating.
Exactly.
So you get it's an evolving population of cells is what a tumor is.
Yeah.
I mean, that's its own area of the literature is modeling that evolutionary process, which is crazy.
And I think
that's well beyond the scope here, Sam.
Oh yeah. I'm not going to talk about Bayesian models of Poisson distributions.
Don't say Bayesian. That's swearing. The thing is, though, let's just imagine we decide to start going through various genes that we know are involved in cancer, one at a time. We could do a hangout of the same length on a single gene. And then there's hundreds of genes involved.
Mm-hmm.
We could talk about this for the rest of our lives and still not have covered it all, because it's such a complex disease, and because every single person who has cancer, their tumor is unique.
It's as unique as they are.
Exactly. And so it's, I said this last week, it's not like an infectious disease like smallpox or whooping cough. You have a single cause and we all come down with the same symptoms. And we can have one cure because it's one causal agent. With cancer, there are so many different causes. There are so many different things going on. And as Sam said, even within one tumor. that trying to get a one-size-fits-all cure is really difficult. And I think a lot of people, they think cancer is a single disease. Or they may have the idea that, well, ovarian cancer is one disease and cervical cancer is another and breast cancer is another. But it doesn't work that way. And the absolute complexities involved in it. It fascinates me. Terrifies me, but it fascinates me. But I want to thank everybody for their contributions. I've really enjoyed just sitting and listening to all of that information. And I think, I mean, from me, a round of applause for our audience.
You guys deserve it.
Yeah, thank you very much.
The questions were great, but they're a little scarce, but that's okay. They were good questions.
You're too good. You're too good at this.
Yeah. Good questions, very thoughtful. But of course, like I said, we've all mentioned this. It's a dense topic. Sometimes you don't even know what to ask when it's your first time through it.
Yeah. And if anybody has been maybe shy of asking any questions in the live chat, you can always send a private G+ message to any of us. And we will answer what we can. Now, before we go off air, though, is there any other big cancer topic that we would maybe like to tackle next week? Here's a thought. Treatments.
I don't know, maybe we could look at.
We could talk about Angelina Jolie for 2 episodes if we wanted to. So
not going to get a protest from me.
Yeah.
And I'll bring pictures.
I mean, there's treatments. I am not hot on treatments. I would definitely want
small. That's not a small section.
It's
you
not.
You have to break it down into treatments for hematology, oncology, treatments for lung cancer. I mean, it would have to be broken down almost into an organ system to be any type of discussion that wouldn't be vastly different when you switch from one organ system to another.
Yeah, that's true. But it might be worthwhile doing one on treatments for some of the commoner cancers like breast cancer, leukemias, that type of thing. Yeah. But I would want to get Hogtie in on that one.
Yeah, I was just about to say, I think that's Hogtie's area of expertise, if I'm not mistaken.
Yeah, I'm less than useless in that discussion.
Yeah, but that would be a good one to do.
Hogtie knows a lot about chemotherapy, or
yeah.
Okay.
Yeah.
Great. Maybe I can learn something from him.
Yeah. Here's another thought, people. Right, we're busy talking about pathologies here. Pathology is the study of disease. And most of us don't even get into pathology until we've done basic anatomy. You know how the system's supposed to work before you start looking at and what happens when it goes wrong. Do you see what I mean?
Yeah.
Yeah, it's trying to correct the stuff that a certain person does. If you're going to, put forth this idea that, well, these masses of cells are somehow absorbing toxins, and that's its function, you might want to kind of know what organ, systems actually do to begin with.
I'm going to drop RSC's link into the live chat. This is for a video on double-strand DNA repair, which is what he was talking about with the BRCA1 and 2. That's it.
Thank you for doing it, Fiona.
No problem. I should have thought to allow people to do that. I never even put cameraman on. Oh boy. Anyway, I'll take us off here now. So, audience, thank you very much. We'll work out what we're going to do for the next one and when we're going to do it, and I'll try and let everybody know in time. And I will attempt to be there when we're supposed to be there, unlike having to cancel last night. But thank you very much, guys. And at RSC, thank you for that. Good night. Say ta-ta, everybody.
Good night, everybody.
Ta-tas. Save them.